So we're talking about multiple alleles and we're talking about codominance. We're talking about the ability to have multiple dominant alleles be present besides the idea of one dominant and one recessive. In the case of blood typing, there are four possible blood types, A, B, and O. And they mean different things based upon what type of proteins exist on the blood cell. If we look at, let's say, a red blood cell, concave blood cell, we're going to learn in cell-to-cell communication later in the year, there's certain proteins that can basically extend beyond, okay, the phosphobilipid layer there that really protects the cell and allows materials to go in and out. And this structure of the proteins allows certain parts of our system to recognize this cell as being either part of us or not part of us, called an antigen. But more importantly, okay, we produce these types, okay, of proteins. Now, of course, we don't produce an A or an AB on the red blood cell, but that's our way of basically coding for and talking about the different type of possibilities. Now, the discussion today is talking about still Mendelian genetics because the crossing of people with A or B or AB or O blood follows Mendelian genetics because these alleles are, I guess, far apart on the same chromosome, or they're on different chromosomes, so the law of segregation, the law of independent assortment, better yet, is followed because of the idea that these are not linked genes, as we'll talk more about later on the year. So our Punnett squares that we can do work out very well in figuring out the relative phenotypes that exist for, let's say, parents that produce certain children with these types of blood typing. But again, the blood type is about the type of protein that exists. And again, it's very important in cell-to-cell communication what that shape is. Now, very, very important that, of course, we have part of our immune system, as we'll talk about, will recognize proteins that are similar that we have on all cells that they'll recognize as being, oh, that's part of our body. And then if there's a protein shape that has changed because it's degraded over time or it's not part of our body, we have certain parts of our immune system will recognize that not to be part of our cells, okay, and attack this cell and kill it. Okay, so we have an important part. So this idea of this important piece of a mechanism that prevents us from either attacking our own cells or attacking cells that don't belong to us, the immunity system, is based upon what kind of proteins exist on the outside of cells. So if you have A blood, okay, A blood, or type A, I should say, you produce a certain shape of a protein outside the red blood cell. Now, if I was to give someone who is B blood, now B blood would mean that your red blood cells have a different shape. Okay, let's make it different. Let's make it look like that. And this shape is recognized by your body to be owned by you, so we don't attack our own blood cell. But if you were to put an A blood type, which is right here, into our bloodstream, okay, our immune system, okay, would attack it and maybe also coagulate it so it sticks together. Maybe there's some antibodies, as we'll talk about, that will link these and have them stick together so that the white blood cells can easily find this package. But this clumping up of blood could block a blood vessel and cause a blockage, which can cause death, stroke if it's in your brain, a heart attack if it's in your heart. So these are very important issues we'll get to. So if you have A blood, you have a different type of what? Protein on the end. And of course, if you've got B, you've got another. Now, if you're AB, you would have a what? A red blood cell that has both of these. That's why it's codominant. You produce both. Okay, you would have the A allele and the B allele that are both dominant being made, so you produce both. So think about that for a second. All right, if I was type B blood, if you give me A, this is something I would recognize as foreign and attack it, and it could clump up if I have antibodies linking it together so that the white blood cells could attack it easily. We'll talk about that. If you put AB into my blood, okay, not good, because I would recognize this part to be foreign, although this would be the same as mine, so that'd be fine, but there'd be this receptor that we recognize to be foreign antigen, and we would attack it. So the only thing a B could have is, guess what? Type B. All right? So it's very important you get that. Now, for the same reasons, if you were a type A blood, okay, same reasons, if you gave it B, we would recognize this to be foreign and attack it, and that could be catastrophic, okay? So if you're A, you can only accept A. If you're B, you can only accept B. Now, if you're AB, okay, now if you're AB, if you could give me A. Now, why can you give me A? Because, well, A is something I already have. This is not foreign. So if you're type AB blood, you could accept A or you could accept B. And the answer is why? Well, you already have these receptors that your body knows is part of our body, it would not attack, and AB has the same. So we call anybody who's AB the universal acceptor. So if you're AB and you need blood, you can take B blood or A blood because these proteins, okay, that cells will communicate with, these are already present in AB, so obviously our body wouldn't attack them because this is something that's normal. Now, what's type O? Type O is a red blood cell that has no proteins in terms of cell-to-cell recognition of blood typing. So it doesn't have any of these types of proteins sticking out. Now, what's the advantage of that? Well, we also call O, well, if you were to try to give type A to O, all right, this A to O, well, the problem is that A has a receptor we would find foreign. If you were to give B to O, the people that have the O blood would find this receptor foreign. If you try to give it AB for the same reasoning, this would be foreign because these types of proteins on the end or on the outside of the cell are not what this cell has, and that would be attacked. So unlike AB, who's the universal acceptor, O is the universal not acceptor, if you think of it that way. But that would all be bad news, so we can't do that. However, because it doesn't have any proteins that recognize it to be a certain type of A, B, okay, or AB, you can actually donate this, okay, to every one of these cells. Think about it. Why could you donate O to A people, or people with blood type A or blood type AB or blood type B? The reason you can do that is because there are no what? Cell proteins. There's no proteins for the body to recognize as foreign. So we call O the universal donor. So if you're type O, you're in high demand because you can use that blood to help give transfusions, or people have low blood amounts, to anybody who is either AB, A, or B. Okay, some background if you didn't already know that. But what are the alleles that make this? This is a great example of codominance because if you have both A and B proteins present, or if you have both what? Chromosomes that have both the A and let's say the B present, guess what? When these chromosomes, okay, are uncoiled as DNA, they can make proteins for the A and the B. Why? Because they're both there. So being codominant just means that, doesn't mean one's better than the other or codominance. It means that both genes are what? They're both working. They're both available for us to make the proteins that make this allele be expressed, or the expression of the gene to be the allele, however we want to think of it. So it's important. Now we have a way that we do the Punnett squares. Just by convention, and don't know the people who thought it was convention, they like to use the I for blood typing. So we have I, and then we have the big A above it, like this, to show that this is the allele and a blood typing for the A protein. So if you look at blood type A, the genotype could be a what? A big A and a little I. Now, a little I means recessive. Now, recessive, as we've already learned, means what? It means a broken gene. The gene's not working anymore. So if you have, guess what? Red blood cells or blood cells that don't have any proteins on the outside, they're not making the proteins because it's a broken gene. So to have an O, what do you need? You need two I's, okay, recessive. If you're an A over here, you would be an I to the A or an I to an little I. That means that this is the recessive, which means it's a broken gene. So you can still express A. This would be heterozygous, or you could be homozygous. And if you're B, same thing. You could be I to the A. I'm sorry, it's B to the B, right, because of B. And then you could be heterozygous, or you could be what? I, and you could be B and IBB, which is homozygous. And of course, to be AB, you would have to be what? Codominant, I guess. All right, you have to have both present. Okay, so the little I thing is just what they use for that. And of course, you can see that here. Okay, so look at this. Blood type A can receive from A because it has the same what? Same protein structure that you would not recognize as foreign because you already have that protein structure. O doesn't have any protein, so there's nothing to recognize as foreign. B, for the same reasoning, has a different protein ending, so you can accept from B because it's the same ending there. And O doesn't have any what? Ending, so you wouldn't recognize it to be foreign. AB would mean you have what? Both the A and the B. So you accept from A because A is something that can't be foreign. You accept from B because B can't be foreign, and AB has both. And of course, O has nothing for you to detect. Okay? Notice O can only accept from O because AB, A or B, both have what? These proteins that are foreign to it. All right, so some background here. So in any case, let's go and do some across here. Please complete a cross between a heterozygous for type A. Okay, so let's do that. So type A heterozygous. Heterozygous, so here's I to the A. And if I'm heterozygous, I'd have the broken gene too. And heterozygous for blood type B. So this would be I to the B. And the broken gene here, the recessive. And what you'd make is you'd make A, B. You would make the heterozygous B, heterozygous A, and then you would get the what? You would get the broken gene, the blood type O here, right, where you're making no proteins. So if you look at your phenotypes, okay, I A and IB is type AB blood. You're making both of these proteins because you're going to express those alleles because they're not broken. Okay, so there's your codominance right there. No one is better than the other because they're both there to do what? Make and code for. So you'll make both proteins. Then you have this one here where you have the heterozygous for A, and you have the broken gene, so you can't make any proteins, but this one still can be coded for and expressed as a protein that will fit on the A, and that's why it's still this is AB and this is A blood. And of course here you're going to have the I and the B and the I. Okay, and this of course would be the phenotype B blood. And of course here with two broken genes, this would have to be, as we talked about, your O. So, okay, so here are some questions. What are the possible phenotypes in the offspring? Well, we have AB, we have A blood, B blood, and then we have type O. So all are possible if you have a heterozygous B blood typer and heterozygous A. Okay, pretty straightforward. We've seen those skills. It's just now you're using blood typing with codominance. Now, let's get to some probability questions, and that's what this is really about besides codominance. What is the fraction of offspring that are O and type A? So what are the fraction? Well, fraction would be the total choices. As you can see, there's 4 possible genotypes, okay, and phenotypes in this case. So what are the fraction that are type O? Well, we have 1/4. And we have AB, another 1/4. And I'm going to add these together. All right, so 1/4 plus 1/4 gives me 1/2. Okay, that should be pretty simple. Now, why did I add? This is important. This was the addition rule of probability. Okay, this is the addition rule. Now, why is it the addition rule? because they asked me something about the same toss. Now, what I mean by the same toss is, if I were to take a six-sided die, okay, if you're playing dice in a game, and this is terribly drawn, but you have the 1, you got the 2, the 3, and you have 6 sides to this, okay? And there are 6 possibilities, aren't there? You can roll a 1, 2, 3, or 4. In fact, the probability of rolling a 1, of course, because it's 6-sided cube, is 1/6, right? Sure. To roll a 1 or a 2, what are the 2 choices? Well, you have 1/6 for the 1 and 1/6 for the 2. You would add them together. If I roll the dice once, what are all the possible outcomes? That's the addition rule. Okay? If I have, one meeting of these two, what? Gamete from the mother, gamete from the father, here are all the possibilities from that one roll. We use the addition rule. Now you'll hear the word mutually exclusive, and you heard about this in your summer assignment. And I said, what does this mean? Well, mutually means that you're tied together. Why are we tied together? We're tied together because we are basically all of the choices of possibility. So if you have all the choices of possibility, in this case, 4 things over 4, you'll always get 1. So as we talk about all the possibilities in one roll or one meeting of the gametes, we would use the addition rule because of the mutually exclusive event. What's exclusive about it? It's based on just this roll, only this roll. What's mutually? Well, because they're tied to each other. These are all the possible choices that can exist. There's only 4 possible choices, so each one of these is a 1/4, or at least each one of these boxes. Sometimes these phenotypes can be the same. So it's a 4 out of 4 kind of deal. So if I wanted to know the probability of being A, B, and B, and A in one roll, in one possible event, that's mutually exclusive. Mutually means it's tied to each other because you're in the one roll. I would add the probabilities. So what's the fraction of offspring from these 2 one-time mating of these gametes to make a zygote could be? That's the addition rule. So the exclusiveness means that it's only from this one event. What's mutually is they're tied together because there's only so many choices. In the case of the dice or the die, there's only 6 different sides, 6 different numbers, and each one is 1/6. So every time you add what the possibilities are in one event, they have to be tied together because there's only 6 choices. In the case of this Punnett square, there was only 4. That's important, and we can see that in our reference tables. So here we are, our AP reference tables, and look what we have here, laws of probability. If A and B are mutually exclusive, then you have the probability of A or B is adding the individual probabilities, and these are the fractions that equal 1. Why do they equal 1? These are all the possibilities in one event that's exclusive from all the other events. Okay? Exclusive means you're in a private resort. It's an exclusive resort. You're one event. So it's mutually tied together because in the one event, we're talking about all the possibilities. And you guys did this in your Punnett Squares over the summer where you talked about, hey, what's the chances of having these couple of phenotypes in a dihybrid cross? And you added all the phenotypes over 16. Okay? So these are all the possibilities in one event. Okay? And if you look at the other one, if A and B are independent events, this would be multiple rolls. Now, if you have what? If I had 2 rolls of the dice, what's the chance of rolling, okay, a 1 and a 1 again? Here I'm going to multiply because now we're talking about these events are individual rolls, okay, meaning we're talking about not an exclusive event. They're inclusive because, well, they're different events, okay? They're not mutually exclusive. They're totally different. So we're meeting gametes another time. So if I said to you, what's the chance of having a boy? You would say, well, because of the X and Y chromosome has an equal chance to go to one side or the other in mitosis, you would say 50%, right? And if I said, what's the chance of having a second boy? Now, if I said to you, what's the chance of having a boy, 50%, or a girl, 50%, that's never going to change. Every individual rolling of the dice is 1/6 for the what? Individual numbers. the chance of having a boy or girl is always going to be 50%. It's an X and Y gene that's spread apart. But if I said to you, what's the chance of having two boys? Now you're talking about two individual roles. It's not adding. You're going to multiply 1/2 by 1/2. And you would get 1/4. And you say, why would I get 1/4? Well, if you have two independent events, what are the chances? You can have a boy or a girl the first time, or a boy or a girl the second time. So those, if you think about all the possibilities, you can have boy, boy, you can have boy, girl, girl, boy, and girl, girl. Okay, so what's the chance of having boy, boy? 1/4. And that's how it works. And how did that happen? You took 1/2 times 1/2. Okay, so that's how you would work that. Okay, and if you didn't quite get that, I'll just show you again. So the multiplication rule, as we just said, is dealing with two different events. So what's the chance of having two boys? Well, we know the first boy is 1/2, and we know the second event, second roll of dice, in this case flipping a coin, boy, girl, I guess more of a flipping of a coin when it comes to the X and Y chromosome, okay, splitting apart, and the chance of you getting that is 50% because you get one or the other. You would times it by 1/2 and you would get 1/4. But what are all the possibilities? What are all the possibilities here, party people? Okay, well, the reason why it's 1/4, and it's the reason why it works, we can do this. We can have a boy-girl combination. We can have a girl-boy combination. We can have a girl-girl, and then we can have a boy-boy. These are all of the what? All the possibilities. And if you notice, boy-boy is what? 1/4 of all the possibilities. So you see how that works. So how do you know you have the addition rule, the multiplication rule? Are we talking about the same role, or are we talking about two or more additional roles? Okay, so that's how that works. Okay, so let's go on to the other questions. What's the probability that the parents will have two children with the blood type AB? So, two children is two different events. They're not mutually exclusive. So what's the chance of having AB? 1/4. And I'm going to times it by another 1/4. Why do I do that? These are two different rolls of the dice. Now the dice that I have is I have a four-sided object where four things can happen. So it's 1 out of 4 possibilities right here. So it's 1/4 times 1/4. 4 times 4 is there's 1/16th chance that I can have two children with AB blood given these two parents with two different roles. Okay, these are independent events. D, what's the probability the offspring will have type A or type B blood? Well, type A, okay, is what? 1/4. And type B is also 1/4. Well, look at the question. What's the probability that the offspring will have type A or type B? So this is in one roll. What's the probability, okay, that I would have roll a 1 or a 2? Right? So these are the same roll. This is the same meeting of the same gametes. What's the probability of just one meeting having a type A or type B? Here I'm adding again. So a 4th plus a 4th is 1/2. This is the addition rule. This is in the same roll. Again, what are all the possible outcomes that are possible with one meeting, with one roll? Okay. D, what's the probability a child would be type O and also be a girl? So what's the chance to be O? Okay, well that would be 1/4 because you have only one II out of a possibility of 4 choices. Now, a girl, well, the girl would be on the sex chromosomes. You'd have to have an X and an X. That's a different chromosome we're going to have to assume than we have where the blood typing. So that's a totally different separating of a different chromosome. So because it's a different roll, a different set of dice, we'd have to times that. And what's the chance of getting a girl? Because there's an X and a Y, okay, chromosome, there's only one of two choices. That's 1/2. And we're multiplying because these are two different events. Why are they two different events? Because you're going to have a splitting of the Y chromosome and the X. The Y is smaller. And you're going to be splitting apart the chromosomes that have what? All right, the A or the B and of course you have the I and the I part of that. So the idea here is that we're dealing with 1/4 of a possibility to get an O from the meeting of these two parents when you split to see which gamete you have. And of course, you're splitting a different chromosome, and therefore it's a 1/2 chance to get a boy or girl. So we multiply two different events. So this would be a 1/8 chance, and that's how that works. What's the probability that, this is F now, that 3 children all have type O blood? Now, if you're having 3 children, can you have 3 children with one mating? I guess you could have triplets, identical, but generally speaking, non-identical triplets, you're going to have 3 different meeting of the what? Meeting of the gametes here. So this is going to be 3 different events. So these are independent events. So to have type O blood, which is, by the way, just 1/4, it's going to be times 3. So 1/4 times 1/4 times 1/4. Okay, 4 times 4 is 16. 16 times 4, and you get 1/64th chance. 3 separate mating of the gametes that come from these two parents, 3 different roles, the multiplication rule. So that's what I'm really teaching you here. I would like you to go on to the next page and do another cross and answer these questions, look at the key, and then I want you to try something really cool. It's called a pedigree diagram. And I want you to see if you can figure out from the pedigree diagram what the other phenotypes of the blood typing would be. For instance, if you look at this one right here, I have a, and again, what you're looking at, the male is the square and the female is a circle. So you can think of this mating of a male and a female. And this is a B, but this B, you don't know if it's a B that is what? Heterozygous or homozygous. Now, if we're going to make O, what does that mean? Somehow there has to be an allele. There's a free I somewhere, correct? If we're going to make I to make O, you can think about then when we do this Punnett square, our parents, the maternal and fraternal, must be what? Have to be to make this. And that's right. They have to be heterozygous. So this would have to be an A, and this would have to be a B. So by knowing I'm making O, which is 1/4 of these are going to be II, the 2 broken genes or the 2 recessive genes, the A or the B has to be the homozygous to have a free I. And if you're going to make O, okay, the question is this an II or is it a B? And you'll have to think that through as you go through this, knowing that you're making an A, B, and an O. So think that through, look at the key. This will be a bit of a struggle a little bit, but the rest of this based on the probability rules that are given to you in your table and putting to use through the addition and multiplication rule is what this was about.